Hello, I'm Dr. BCMR. We have seen that mutations occur, that is, change is normal. Change in the sequence of the nitrogenous bases is mutation. And most of the mutations are preceded by DNA damage. And damage can be many types and many forms, and how that damage translates into a mutation and how these mutations are repaired is the subject of what we will talk, at least partially. The mutations could, or the DNA damage could be something like loss of the whole base or functional groups or some binding between or chemical covalent bonds forming between the bases, like as in the thymidine dimers. Or it could be mismatches that are usually caused because of replication, errors during replication, or it could also be because of homologous recombination when there are differences between two homologous regions. There are also groups that could be added on to the nitrogenous bases, something like methyl groups or ethyl groups, or oxidation of the bases themselves. So one thing is central, how a DNA damage becomes a mutation, is the step of replication in between. So if there is a damage, and because of those damages, there could be mispairing or the base pairing. What normally would be A always base pairs with T and G always base pairs with C. But that doesn't have to be the case in some cases. It's because if there is any chemical change within the nitrogenous bases, it might affect the base pairing capacities, and then that would alter many other things. We'll take an example of one of the nitrogenous bases, that is with guanine, and see what all could happen to it. Just one base could happen. Now we have 4 different bases, and each of them could be affected as well. So first thing is hydrolytic damage that could happen because of reactions, and we might have the glycosidic bond breakage, because of which the base could have been completely lost. This is true with almost all the bases. At the same time, we also have several other positions that could be affected. Something like this bond could be, oxidation of this bond could happen at 8 of it. So we will end up forming oxo G, which can base, because of which the base pairing capacity, normally guanine only base pairs with cytosine, but now because of the oxidation of the guanine, its base pairing can also base pair with adenine. So that is, if replication were to occur and we have an oxo G, then it might be base pair with adenine or cytosine. We should remember that the DNA polymerase, when it is polymerizing, it can only check the validity or the correctness of the incoming nucleotide, not of the template. It cannot do anything to the template. So that is why what was a DNA damage, like the oxidation of this guanine, and it could become, because through replication it could become a mutation. And the compounds within the cell that are generated as byproducts of metabolism, like reactive oxygen species like a nascent oxygen H2O2 or OH-, they could cause this oxidation. Similarly, alkylation can also happen. Alkylation is addition of ethyl or methyl groups onto different positions. So alkylation of the position here, 6th carbon, could cause mispairing. Something like now guanine, which normally base pairs with cytosine with 3 hydrogen bonds, is now can base pair with thymine with 2 hydrogen bonds, and that will, upon replication, could become a mutation. So deamination is also quite common, and the same thing can be with even with adenine, like we have seen in the previous example. So alkylation, when we speak, the compounds, something like induced nitrosamines, they could cause alkylation of the nitrogenous bases, more specifically guanine here. So there are simple ways of how you can get rid of stuff. So DNA or the evolution has come up with ways to retain the DNA as it is, or it is kind of reversing the damage. Those kinds of repair mechanisms are referred to as direct reversal. Say, for example, in this case of alkylation, assume that there was an alkylation has happened in this position, because of which mispairing G is base pairing with T, and there are enzymes called as methyl transferases which bind to these alkylated nitrogenous bases and transfer upon to themselves the methyl group, for example, and then go get degraded. Of course, this is a very expensive one, because to synthesize methyl transferase, the cell would have spent some energy and resources. Now, just because it removed one methyl group, it is after the transfer of methyl group onto itself, the whole enzyme is going to get degraded. It's a very expensive process or way, but that is worth it, because mutations usually are detrimental to the cell. So typically in biological system, it's not about efficiency, but it is about remaining as such, existing. And that's why it is worth the price to pay, to keep to undo the damage that has happened, something like that of alkylation. This is an example for direct reversal. There is also another one, another mechanism which is also an example of direct reversal. Say, upon ionizing radiation or say UV light, there are thymidine dimers that form. That is, there is a cyclobutane ring that forms because of the energy, the carbons here, they form this cyclobutane ring, and now the base pairing capacities are influenced or affected. That is caused by UV light. You call these as thymidine dimers. And if there is a thymidine dimer in one location, and now if that particular strand happens to be the template for transcription, RNA polymerase doesn't know what to do. Same is the case if during replication. DNA polymerase will get stuck there because it doesn't know what bases to add, because the bases, the thymidine are base paired, are formed a cyclobutane ring, because of which base pairing is impaired. So the effects will be serious. Say, for example, the same thing happened in a big or an important gene, then both transcription and translation are affected. So there is an enzyme called as Photolyse, which is a very efficient one compared to that of methyltransferases we just discussed. That Photolyse is an enzyme which recognizes these thymidine dimers, and using visible light, it takes the energy from the visible light and uses that energy to break this cyclobutane ring. So it is just a reversal of what UV light has done. It is just a reversal of what UV light has done. It is another example of direct reversal. And compared to methyltransferase, this is much more efficient, because all that Photolyse does is after exposure, and if we let it stay for light in the dark for some time and then put it in the light, during dark time it will scan through the DNA, bind to these thymidine dimers, and using the visible light it will undo the cyclobutane ring. And now DNA polymerase can read these bases, and so do RNA polymerase, so that replication can be affected. Translation is not, it should have been replication. I hope we had a fair enough idea about what all things happen. These are direct. There are also many other, as I said, the mismatches may happen because of replication errors. And if there is an abasic site, what to do with it? We have to fill up the gap somehow, otherwise replication and transcription are affected. So there are specialized mechanisms for them. We'll check it out later. Thank you for watching. Bye.